Thank you. So good afternoon, everybody, and welcome to the Alberta Transplantation Ste seminar today. Just a couple of acknowledgements before we get started. I'd like to acknowledge the sponsorship of the Paladin for their support of the ATI seminar series. I'd like to acknowledge the University of Alberta that we are located on Treaty Territory 6 and respects the histories and languages and cultures of First Nations, Métis, Inuit, and all First Peoples of Canada. This presence continues to enrich our vibrant community. We have a wonderful speaker today, Dr. Rogers from Toronto. Dr. Rogers is an associate professor at the Lunenfeld-Tenenbaum Research Institute at the University of Toronto. He's also an associate scientist at the Jumeirah Transplant Center at University Hospital Network. He completed an MSc in immunology and a PhD in embryo development with a focus on granulation at the University of Toronto. During his postdoc with Dr. Robert Casper at the University of Toronto, Dr. Rogers focused on advancing umbilical cord blood stem cells for hematopoietic stem cell transplantation. Dr. Rogers, along with Dr. Casper, founded the Inception Life Bank, the first and largest cord blood program in Canada, and is currently acting as the scientific advisor at the Inception Life Bank. Dr. Rogers studies hematopoietic stem cells and their use in regenerative medicine. His research focuses on stem cell therapies for peripheral vascular diseases, non-healing diabetic skin wounds, and kidney disease. He's developing tissue culture systems using decellularized organs such as kidney and ovary and pancreas. In 2020, Dr. Rogers received CTRDP Research Innovation Grants for his project, Building Matched Organs Using Patient-Derived Stem Cells. So thank you so much, Dr. Rogers, for giving the talk today. Over to you.
Thank you. So let me just share my screen. So today I'm not going to talk about anything with umbilical cord blood at all. So can you see my screen?
Yes.
Okay. So first of all, thank you for the opportunity today to talk about my work. The title of my talk is The Development of an Ex Vivo Whole Organ Perfusion System for Donor Organ Studies and de Novo Organ Production, or the short title is How to Make a Kidney. So let me just make sure my slides will advance. Yeah. So this is an overview of what we're doing in the lab. I apologize. I'm just going to minimize the screen for myself here so I can see what I'm doing. Okay, so this is an overview of what we're doing in the lab, and our ultimate goal, as the title stated, was really to humanize a whole pig organ so we can make donor organs. And in order to get there, there's a lot of other work we need to do in order to fulfill this goal of making human donor organs. So I'm going to basically walk through all the work we've done in the lab in the last few years and what we're working on now. Okay. So I want to start with really the bioreactor development using native kidneys in an ex vivo perfusion system. And that this is important because if we want to make a kidney, we have to be able to grow it. We have to be able to maintain it ex vivo. These are just some of the different bioreactors that we've used and we've worked on in the past. So when we're developing a bioreactor to actually house a kidney, and this is not a trivial endeavor. There are commercial units for maintaining rodent organs, for example, but they're usually used for 24, maybe 48 hours tops. They're all done on the bench. They're not very sterile. And they're not really designed for long term. And we really need to be able to grow our kidneys for weeks, if not months. And I'll explain the reasons why as I go along. So, really we're looking for developing a system where, you have a heater to keep the perfusate at 37 degrees. You need an aerator in order to oxygenate the system. You need a couple pumps to keep all this going. You have water jacketed tubing. So these systems, as you can see here, are very cumbersome, but they work. So when we first started doing this, we needed to be a bit innovative because, generally you have an idea, but you don't get grant money necessarily until you have some preliminary data. So we started off by taking falcon tubes, 50 ml falcon tubes. They're 50 cents each, and basically hacking them so we can make little bioreactors and grow the kidneys in the bottom. And then we kind of moved in where we bought some of the commercial parts of the commercial units that were available, but we took these little Corning jars, drilled holes in the top, and connected our kidneys. You can see there's a little mouse kidney in there, and we ran these systems. And we found that they were very difficult to control. And as I mentioned, they weren't really designed for long-term. So for me and for my lab, we needed to start working with engineers. So it was interesting because having biologists is great. A lot of people in the lab were PhDs or MDs, but we needed to bring in people with engineering degrees. And I had different engineering students come in the lab, and these were actually undergrads, and they were great, very, very smart students. So the work I just want to touch on here was one student, Natalie Wan, who came in as a first-year summer student after her first year of bioengineering at McMaster University, and then she came back for the second summer. And she did a lot of mathematical modeling on the bioreactors. And her goal was, could she design something that was small, that didn't require an aerator, because you get enough passive diffusion, and something that was easy to work with. But then also for our later experiments, when we're going to recell and decell the kidney, or decell and recell, we need to put the kidney under vacuum pressure. So the bioreactor needs to withstand vacuum. So Natalie came up with this design, and we took her design, and based on the mathematical modeling, we actually had it built. And then we did validations of this. And I'll show you some of that data. So you can see here, we have a mouse kidney hooked up to the bioreactor that Natalie designed. And in this case, you can see we have the artery and the vein is cannulated. And this is actually pretty tricky to do because the mouse kidney is so small. We don't want to move to rat or rabbit kidney because, and I'll explain a bit more in a minute, but there's a lot of genetic knockout disease models in mouse. And we want to be able to take advantage of those models for future disease modeling in the system where we're going to use isolated organs. So the other thing that we're able to do, and I'll show you some data from an experiment that we did, we can also cannulate the artery and we can cannulate the ureter. So if we want to actually collect urine from these kidneys to see if they're functioning, we're able to do that also. So with Natalie's design, her math was great because she calculated that the surface area of the media in the bioreactor with the amount of media we were using allowed for enough passive diffusion of atmospheric air that we could actually get enough dissolved oxygen in the media. We didn't need an external oxygenator. And then this unit was small enough that with these small little pumps that are sealed, we could put this whole thing in the incubator. And now we didn't need water jacketing tubing. We didn't need a heater. So we've gone from that complicated unit you saw a couple slides back to this very basic unit. And this was very important. So I'll just go back to this slide. This was very important because we want to do a high throughput analysis. And you can see here that if we are running studies where we don't need the vein or the ureter cannulated. We can actually hook up 2 kidneys, and we've now modified our bioreactors where we can hook up 4 kidneys, and we have 4 of these, so we can actually run 16 kidneys at once. So this is very important because it just allows us to gain a lot of data very quickly. We're also do a lot of work with Dr. Mark Kesselsner at UHN. And as I had mentioned, our goal is to ultimately make a human donor kidney. And the idea there is to take a pig kidney, strip off the pig cells, and put on human cells. And again, for proof of principle, proof of concept, we made sort of homemade bioreactors. So this is, I call it my Canadian Tire special, because I went to Canadian Tire and bought a Tupperware container, bought an automotive gauge in order to monitor a vacuum pressure and positive pressure in the system. $25 worked really well, but it wasn't sterilizable, but it did at least allow us to gain some knowledge. And then just like in the mouse, our next step was, to make a system that was sterilizable. And instead of making a glass and metal system, we did try some other alternatives, again, thinking about high throughput and trying to keep the costs down in the long run. So we would take these tissue culture bags and modify them. I'd basically cut open the bottom, turn them inside out, be able to put in these adapters, and then put the kidney in and then seal it with a heat sealer. And you can see here that we have our, our cannula connected here for the artery, and it's water jacketed to keep it warm. We have the venous outflow here, which goes back to the reservoir where it's warmed and then put back. And then we have this 3-way spigot here, essentially hooked up to the ureter, so we could sample the urine when we needed it. And then just like in the mouse, you can see here that, again, it's on the bench. We have to use our water jacketed heater. We have to have an oxygenator. It actually takes 3 pumps to run this. And it was very difficult to regulate the temperature actually and the whole system. So again, I had some engineering students come in, and they started to design a pig bioreactor that could withstand, vacuum pressure or positive pressure, which was required when we go to put cells in. You can see this is quite a big unit with the pig kidney sitting at the bottom here. But again, it worked, although we had trouble getting it in the incubator. And then Natalie came back the next summer, and building on the data we had and building on her mouse work, what she was able to do was make this nice compact porcine kidney, or really porcine organ, because we use this for more than just kidneys, bioreactor. And this is important. Again, she did mathematical modeling and showed that because the pig kidney consumes so much oxygen, that the surface area was not large enough to allow for passive diffusion, that we do need an external oxygenator. But trying to stick with the ability to make this simple and easy to use for everybody, and we would like other labs to do some of the work that we're doing. We'd like to be able to share this knowledge and make it as easy for them as possible. Natalie found some papers dating back to the 1970s where groups were trying to make what they call artificial lungs. And essentially what they were doing is they were taking silicone tubing, and the silicone tubing it's permeable to gases. It allows for oxygen from the atmosphere to diffuse in. And so you run your media through this tubing before it goes to the kidney. This increases the surface area. And Natalie calculated that using different lengths of tubing, you're going to get so much diffusion. And then we actually did the actual experiment and showed that if we hook up about 6 meters of tubing, we get enough surface area to actually properly oxygenate the pig kidney. And the nice thing with this, it's the same principles of the other systems we used, but it's all, again, self-contained in the one bioreactor. And we can just take this with the same pumps and just put this right into an incubator where the temperature is regulated much better. We don't have to worry about all this external equipment. And again, this is very key for doing high throughput experiments and for not having to take up a big footprint in the lab and trying to keep the sterile by having it outside of a tissue culture hood, for example. So what I want to do is just show you some data we have with our mouse kidney test, and this is with a native mouse kidney hooked up to our bioreactor. And we use an oxygen sensor at the artery and another one at the vein in order to monitor oxygen. And when we first hook up the system at the very beginning, you can see that the oxygen level's about 6 parts per million. It seems to spike a bit, and then it comes down after 2 to 3 hours, and it stabilizes at about 5.8 parts per million. And this is sufficient because the mouse kidney will only consume about 1.5 parts per million. So you can see at the vein that even though it does fluctuate a bit, it never drops to 0. So this told us there's always sufficient oxygen in the system. And again, Natalie's calculations were perfect in that we don't need an external oxygenator for the mouse kidney bioreactor. And you can see here that this is the end of a 24-hour experiment where we took the kidney off at this point. And obviously the oxygen level goes back up when there's no kidney in there consuming oxygen. So this was due to the kidney, not due to some other reason. This might be a little hard to see, but hopefully you can see it on your screens. We did histology on the kidneys because we wanted to see how long could we grow these kidneys. Again, for some of the studies we want to do, we really want to be able to grow the kidneys for weeks, if not months, as I said. And this is a big ask because the current systems, as I said, for mouse kidneys, but even the ex vivo perfusion machine they're using for pig kidneys at UHN, 24, 48 hours seems to be the maximum. And one thing to keep in mind, I mean, we have to look at our model system, is that the mouse kidney is probably a little easier, a little hardier than an adult human or an adult pig kidney is. So not all of this will perfectly translate from our mouse studies to our pig studies. But again, with the mouse, it's a lot easier to run 10 or 12 mouse kidneys than it is 10 or 12 pig or human kidneys. So we can gain a lot of data from this work, and then we will translate it to our pig studies, realizing that there's going to be some caveats and some new hurdles we'll have to deal with. So one of the first experiments we did was just test different media. So we got a renal epithelial cell media. It's a specialized media for growing renal epithelial cells in a monolayer culture. And we thought this would be better than using a 10% FBS DMEM. So we wanted to compare the two. And this is our day 0 controls at different magnifications. And you can see the tubules, you can see the nuclei, you can see the glom. So very healthy, obviously, time 0 kidney. And then at day 4 in the REC media, day 7 and day 9 at the REC media, there really isn't any difference. And we had the pathologist at our animal facility analyze these slides for us. And she was able to verify that they look healthy. We did the same with the 10% FBS DMEM, day 4 and day 9. And sorry, I'm going to go back. You can't really see much of a difference at day 9, but the pathologist did point out that she was starting to see a bit of tissue damage, just slightly. But that told us, obviously, that the specialized media is better, as we expected. We also did some immunofluorescence in order to look at the morphology and the physiology, anatomy of these kidneys. So you could see again the day zeros, we stained with LTL and KSP, which marks tubules. We also did LTL and E-cadherin, which is another way to look at proximal and distal tubules in the kidney. Aquaporin will highlight the collecting ducts. And then we did nephrin with LTL, and the nephrin highlights the podocytes and gloms. And again, this is day 0, day 4 REC, day 7 REC media, day 9, and then the 2 days with the FBS media. And when you look at these, obviously immunofluorescence is not quantitative, but when we look at the gloms, especially with the nephrin, that we couldn't really see any difference between the day 9 kidneys and the day 0 kidneys. So again, this really told us, this data, that our bioreactor was working well, and with the REC media, we could get about day 9 data from the kidney. Now, as I mentioned before, you know, we have a choice. We can cannulate the artery and the vein, or the artery and the ureter. And it's actually very difficult to cannulate the ureter in a mouse because it's very fragile. So you have to be very careful when you're tying in the cannula with the suture not to damage it. So we were able to get data for a few mice at day 4 and day 7. And I'm just going to show you the day 7 data here. So again, I said it's urine, but it's really ultrafiltrate. It's what's coming out of the ureter. It's not true urine because it's in an isolated mouse kidney. So there's we cannot assess all the criteria of a normal urine sample. But what we did with the animals, with the kidneys, we changed the media every 24 hours. So in this case, at day 7, we changed the media from the REC media back to the FBS DMEM high glucose because we wanted to see what would happen with high glucose. We left it in the kidney for 20 hours, and then we sampled the urine, or the ultrafiltrate, and you could see that the perfusate, which is the FBS DMEM, has very high total protein, 386 mg per deciliter, over 3 mg per deciliter of albumin, and 450 mg of glucose per deciliter. But the urine, the ultrafiltrate, had low levels of total protein, very low levels of albumin, which told us that the albumin was being taken up by the kidney. The kidney was functioning, and the same with the glucose. So this was strongly indicative that at day 7, the kidneys not only look good, they're actually functioning to the best of our ability of being able to test function. Before I get to the next step, so really what the bottom line for this work is that we have a bioreactor that we can use, that when we start to make new kidneys, and if they fail, we know it's not because of the bioreactor, it's because of some other possibilities. So it was key to have a good bioreactor. I don't have the data for it, but we're now up to day 21 with the mouse kidney. And we've also have connected a mouse pancreas and kept it alive for 4 days. And we were able to get a positive glucose tolerance test from a pancreas that was connected for 4 days. So we're working on a number of different organs, and we're trying longer times. Okay. So part 2, and I hope I'm not talking too fast. And if I am, someone can just unmute and tell me to slow down, but I want to try to cover everything. So the next step in our goal of making an organ for a donor down the road is we need a substrate. We need a 3-dimensional substrate to put the donor stem cells on. And the obvious thing to do for us is to decellularize a whole kidney. So that's what we actually started to do. We do decell recell. And then the big question was, once you strip off all the pig cells, for example, you want to put human cells on. And ideally, you want it to be a match to the patient. And ideally, you want to use cells that have come from that patient. And the big question in our group, and not just my group, but there's a larger group of us that are working on pancreas, kidney, and lung decellularization, recellularization, is what type of cells do you put on? If you put on adult cells, adult kidney cells, will they self-organize somehow? Or will they just land randomly, be happy and healthy, but not really give you an organized tissue? And my group, we put forth a hypothesis that the best cells to use would be a progenitor cell, is to come up with one cell type that can make every kidney cell that you require and put that on. And then the idea would be that the extracellular matrix under those cells would then tell it what to be. And, this wasn't just wishful thinking. This is based on actual published data. So we recognize that the biological pathways to use during organogenesis or organ development during fetal development are maintained by the adult for tissue regeneration. So nature basically, instead of reinventing a whole new system for fixing adult tissues, it just said, hey, what we use for making the organ in the first place is good enough for tissue regeneration or repairing that organ in an adult. And then lucky for us, a lot of these important growth factors are actually maintained in the ECM, in the extracellular matrix. So the idea was, if we can remove the cells, but we maintain the ECM, then we have the two things going for us. We have the highly organized structure of the extracellular matrix of an organ, but we also will retain this biological activity. So because of the conservation of bioprocesses between organogenesis and adult organ repair, the adult ECM will actually direct the differentiation of stem progenitor cell types. So in a sense, it's sort of a self-organizing system. So that's great. The literature suggests this should work, and so it's time to put it to the test. Okay. So in the lab, we have focused a lot on stem cell production, on making induced pluripotent stem cells from adult cells. And in my lab and with collaborators, we've collected urine samples where we've taken out, been able to collect the few cells, live cells found in a urine sample and make induced pluripotent stem cells from a patient that way. We use umbilical cord blood cells, and we've also made iPS cells from umbilical cord tissue cells. And it's the system we use is an episomal system. So the episomes carry the reprogramming genes, the ones that Dr. Yamanaka used in his initial paper in 2006. It's a 21-day process. You can get some very nice iPS cells from the episomal system. And the other reason we use the episomal system, it's the same system that Dr. Yamanaka is using in Japan to generate iPS cells for his clinical trials. So we wanted to follow a system that already has clinical trial approval in Japan. It's easier to translate that back to Health Canada. So we can make these nice stem cells that are derived from the patient. They express NANOG, which is indicative of a pluripotent cell, also SOX2 and OCT4. For most of our work, we are using the line we derive from cord blood, just because it works very well. So now we will take those cells and we'll try to prove our hypothesis. So this is our decellularization system. It's very simple. We pump through a mild SDS detergent, and over 24 hours, this is a mouse kidney, it'll take a normal mouse kidney and it'll decellularize it. And you can see here in the bottom, I'm hoping you can see my arrow. that you can see here. This is a normal mouse kidney. It's not decellularized. You can see all the tubules. And then this is the kidney once we strip off all the cells. You can see very nicely the ECM framework is there for the new cells to go on. One of the things that was very interesting in the literature for us is that a lot of groups before us who were doing decellularization, their focus was very strongly on just cleaning up the ECM as well as possible, getting rid of all the cells and the cell debris. And they would use high levels of SDS. And sometimes they would decell for up to 2 weeks at high pressures in order to keep it clean. But what was happening is they were, even though the ECM looked pretty good, they were doing a lot of damage to the extracellular matrix, and they were stripping off a lot of proteins. And back then they were thinking that the ECM was just like a frame, a structural frame. They weren't worried about its biological activity, so that didn't really matter. In our case, it's critical that these growth factors are retained on the ECM after we strip away the cells. And that we don't do any damage. So this is just an example of where we did corrosion casting. And this is we decell, and then through the ureter, we pump through basically a liquid plastic that will harden, and then we dissolve away the extracellular matrix, and it allows us to look at structure. And you can see the capillary tufts that make up part of the gloms, and you can see that there's no damage to them. We didn't blow them out when we were pumping our solution through it, or else we wouldn't see these structures. So that was good. It told us that we were being gentle enough. And then when we did scanning EM at high magnification, you can see the tubules are intact. There's no tearing. There's no physical damage to them. And then this picture I like is it's, again, scanning EM, where we took a cross section through glomerulus. And you can see here that this is not due to damage from decell, it's due to the sectioning that we did, and you can see how thin the ECM is, but it's all intact. And again, this is very, very important because you don't want to damage the ECM. You need a nice substrate for putting your cells back on. The next step for us was to do this for pig. And again, it's a matter of scale up. So instead of using hundreds of mils of media, we use hundreds of liters of SDS solution to decellularize the pig. We get the same nice clean substrate. You can see an H&E of a cross-section of a native pig kidney and then a decellularized. And for our studies, we even were able to get some human fetal kidneys, which we decellularized and try to recellularize, because we really wanted to compare to see whether a fetal kidney was better. It wouldn't help us clinically, but we just wanted to see as part of our hypothesis of whether a fetal ECM was similar to an adult ECM at driving differentiation. Okay, so again, as I mentioned previously, a lot of groups found that, you know, just pump in some solution, don't worry about if you're doing any damage or stripping off any cells, let's just get a clean ECM. And then we wanted to avoid that. So a student in my lab, Tanya, a PhD student, she set out to optimize the decellularization process. And one of the things she did in order to get a lot of data quickly, because she was focusing now on the pig kidney, is instead of using whole pig kidneys, which are difficult to do, and you don't get that many of them, believe it or not, that she would do sections of tissue. She would do biopsy punches, and then she would slice these punches, and she could analyze 24, 48 sections at a time. So it allowed her to do a lot of data, generate a lot of data. And what she was focused on was using low amounts of SDS at below what's called the critical micelle concentration. And the critical micelle concentration is important because Above that, what happens is that the SDS will actually form bubbles around the protein and form bonds with the protein that are stronger than the bonds between protein to protein interactions, and essentially separate the proteins from each other, lift them from the ECM, and they get washed away. And what's important here is that we still, even in the low amounts of SDS, we get nice decellularization, which is good. But importantly, we retain some of the critical proteins like the GAGs. And you can see here that this is a whole kidney 100%. And then when we use 0.05% SDS, we retain about 60% of the ECM proteins, and then it drops off as your SDS gets higher. And the reason this is important is the GAGs, or the glycosaminoglycans. You can see here, they're really the link between the important growth factors we want and glycoproteins, and then their connection to the ECM. So if we can show we're retaining these, and we know we're retaining everything else. And that's why this graph is actually critically important for the work that we're doing. We also did mass spec to measure extracellular matrix protein retention. And here you can see 1%, which is above the critical micelle concentration, 0.05%, which is our target, and then slightly higher. And you can see there's a lot more proteins retained with the 0.05% SDS. And then when we started looking at growth factors, specifically at growth factors, which are the ones we're really interested in, and the 0.05% SDS decellularization is in blue. And you can see that with all the growth factors we looked at, that in the vast majority of the cases, we have the higher retention with the lowest amount of SDS. So the nice thing here is we can get a fairly clean substrate free of cell debris, but we're not really damaging the SDS. So, you know, that was 4 years of Tanya's life working on that. And so now, you know, we have our bioreactor, we have optimized the decellularization, you know, we have our method of making iPS cells from our patients. So what we want to do now is put it all together. So we wanted to make sure that our iPS cells actually can make kidney cells. So we made kidney organoids. So this has nothing to do with the ECM. We just wanted to see if we can make organoids. So we found that in our organoids, we have endothelial cells, tubule cells, and podocytes, and that they will make very nice kidney structures in these organoids. So we know our cells are doing what they're supposed to. There was the ECM is highly conserved between species, but we did want to check to make sure that human cells would grow on a pig ECM. So we did a series of studies where we grew mouse cells on a decellularized mouse kidney ECM, human cells on mouse cells on pig ECM, human cells on pig, and they do survive quite well. And we also did fetal and adult. This is lung, so I'm just putting this in here because at the same time we were doing kidney, we were also collaborating with Martin Post at Sick Kids in Toronto on similar lung studies. And his student, Sherry Chauget, she took rat lung, decellularized it. You can see it here. She recellularized it and got very good recellularization with lung progenitor cells made from mouse embryonic stem cells, and that they matured quite nicely, and she would get these nice mature ciliated cells. And the lung is actually not taking away from Sherry's data, but much easier to work with because it's not as dense as the kidney. So it's a little easier to decellularize, and it's easier to recellularize. So we were able to progress faster in the lung than we were actually in the kidney. This is the kidney work, the initial kidney work that we did. And in this case, we didn't use human cells or pig yet. We took mouse ES cells and put them on an acellular mouse kidney ECM. And just to prove our hypothesis. And if you look down here to E, this is a native mouse kidney, a cross section, and we stained it with cytokeratin, which highlights tubules. So you can see there's a tubule there and there's another tubule there. And then when you look at F, which is our rebuilt kidney, you can see that we get very similar tubule structures throughout. So our rebuilt looks very similar to a native kidney. And then when you look at a low magnification, you can see that we get new tubules throughout. We also stain for KSP, which is another tubule marker, and H is the native kidney. So you can see the tubules here. You can see our rebuilt kidney. And one of the reasons I like to show this is mainly for transparency. You can see that even though we get nice recellularization and differentiation, we experience some damage in the kidney. So we're now trying to balance getting the cells throughout the kidney, but not forcing them in so we don't actually physically damage the ECM. But this is an area we're working on. And then down here, if you look at J, the blue is DAPI stain, and you can see there are cells throughout this section of the kidney. And again, for this D-cell recell, we put on a kidney progenitor cell that should make all of the cells of a kidney that we need. And with the tubule, it looks like the ECM is directing differentiation. And here what we did is we stained this section with WIT1, which marks the podocytes. And you can see that the progenitor cells that landed where a podocyte should be, the underlying ECM basically directed it to become a podocyte. So again, we can put on one cell type and get sort of like a self-organizing system here where we can start to build a kidney. So based on that work, Teresa Chow, who is a PhD student, she did the same, but now with acellular porcine kidney ECM and human iPS cells. Where she differentiated them in a dish to kidney progenitor cells and then put them on sections of pig kidney. And you can see here we get nice proximal tubules, we get endothelial cells, we get nice podocytes. So this is obviously an optimized picture in that we scanned the section for an area that looked beautiful so I could show you and impress you. But again, one of the areas we are now working on is trying to get 100% of the kidney recellularized. So it's more of a technical issue. Up to this point, we were more focused on proving our hypothesis that using progenitor cells is the best way to go. And just before I'll take up just a few more minutes to show you the last part, but just before I get there, I'm not showing you this data, but Tanya did some work where she did test adult cells. So we grew up adult kidney renal epithelial cells. She recellularized pig ECM. The cells grew really, really nicely, maintained all their renal markers up to 35 days, but they didn't self-organize. So what we were thinking might happen is that these cells are already mature. They're not going to need to be differentiated, but maybe they would migrate to the right spot in the kidney and they would self-organize that way. And it turns out that that doesn't happen. They don't migrate. They're happy, but it's a disorganized system. So again, this emphasizes that we really need to focus on the progenitor cell system. Okay, so at the end of the day, our focus is still to make a donor organ. And one of the important things in this is that we want to see if we can run a system from making iPS cells from a patient, decellularizing an organ, growing up everything we need, growing the organ, and then producing a kidney, and showing that it functions. So it's important at the end of the day that it just doesn't look good, that it actually functions. So just in quick summary, we did actually run 2 pig kidneys through this whole what I call system where we're trying to build a whole kidney. And we did this to look for bottlenecks and problems, and it's also just to work out the logistics. So we were able to decellularize some pig kidneys. We did take patient iPS cells. we grew up a ton of cells. We actually would grow up, our aim was to put in about anywhere from 500 million to 1 billion cells we estimated we would need to repopulate a kidney. So we use these Corning cell factories that allow us to grow up large quantities of cells. Then we did our recellularization and our decell resell, and then we grew it in our bioreactor. And then at the end of a couple weeks, and we knew that this might be a bit short, the time might be too short to allow for full differentiation, we took it off anyways, and we thought, well, we'll run it to see what happens when we hook it up to the ex vivo organ perfusion machine that Dr. Selzner uses to assess human donor organs for his transplants, because we wanted to see if our rebuilt kidney would actually work on that system. So just a couple of quick slides. This is Dr. Selzner's group cannulating the artery, vein, and ureter of one of our rebuilt kidneys. And then this is the kidney hooked up to a system. And I'll just run this little movie. Hopefully you can see that. And you can see at the top here, this white thing is the ureter is cannulated. The kidney pinks up quite nicely. The perfusate will go through it. There's no blockages. If it's going to make urine, you'll see the urine will come through the ureter at the top. And you can see that it'll actually start making urine. It'll start dripping on his foot there. He realized that there is urine being made. And you can see, I don't know if you can see it, but it is being collected. Now, this kidney, like the first one, this is the second one we did, only lasted 15 minutes. And what happened is it got a clot and started to expand, and I thought it was going to blow up, so we stopped the experiment. But even though this experiment only lasted 15 minutes and the kidney was not fully recellularized, it told us a lot. It told us that we have to recellularize the whole kidney, because in this system, red blood cells are used to help with oxygenation, and they start to bind to the bare ECM and form clots. The nice thing is that we did get some urine production, and even though the urine is pretty awful looking, that you can see that when we sample the perfusate, you can see there's red blood cells, but when we sampled the urine, even though it's red, there are no red blood cells. So it told us that after all the manipulations we did in preparing this kidney, the glomeruli basement membrane was still intact. So overall, we still have a long way to go. So two areas that we're looking at, and this is my last slide, is that we do understand now from the work we've done, it may actually take months of ex vivo incubation to mature our kidneys from progenitor cells. And it may actually not be possible to maintain a kidney on a bioreactor for 2 or 3 months. And our solution to this is to transplant an immature rebuilt kidney into a patient and let it mature while the patient continues to be on dialysis until that kidney becomes functional. And really, this strategy comes from what they're doing currently with stem cell derived islets. So when you make islets to treat type 1 diabetes, it actually takes 9 months in the body for those islets to mature. So what they're doing in their clinical trials are transplanting these stem cell derived islets. The patient continues to monitor their glucose. They will give themselves insulin injections, and then as the islets mature and start to function, they find they have to give themselves fewer and fewer injections till the point that they don't have to do that. So that's kind of the idea we're leaning towards. The other thing is that even though the extracellular matrix is highly conserved, and we actually did studies where we would transplant human kidney ECM into a mouse and show that it was not rejected because it's so highly conserved, you still ideally would like to get rid of the pig component. and the ECM is turned over constantly, that we're now running studies to figure out how long does it take for the human cells on the pig ECM to actually replace it through natural turnover, resulting in a 100% human kidney. And this time will be what's critical for how long we keep the kidney cultured before we would consider transplanting it into a person. So I just want to recognize these are all the current people in my lab who are doing work on the decellularization of kidney, pancreas, or lung. These are past people in the lab, and then the engineering students. And we've also worked very closely with Dr. Selzner and Tom Waddell. And then we do have a collaboration with AstraZeneca, and these are all the funding agencies that have worked or helped fund this work.
Perhaps while people are wanting to ask their questions, maybe I just ask very quickly. Dr. Rogers, so with regards to the flow rates that you're using for the bioreactor, what impact does that have on the differentiation of the stem cells?
Yeah, that's an excellent question. So we know from our organoid studies that you need flow. It's important for differentiation, and especially for the blood vessel formation. So when we were using the sections, we discovered, because it's a static culture, there's no flow, the differentiation isn't as good as when we recellularize a whole organ and have actual vascular flow. So we know it's important, but we haven't looked, actually measured flow rate and pressure yet. So the engineering group is helping us set up basically sensors for flow rate and shear force and pressure, so we can actually measure and start comparing that, because we know it's important. So that's an excellent question.
Thank you. Yes. Dr. Laurie West?
Hi, yes, Ian, that was a terrific talk. It's really an interesting area. I wonder, Doris Taylor in Texas, you may be aware of her work, mostly in heart, I think. I think she's worked almost entirely in heart, but looking at a similar decellularization and then recellularization strategy. I just wonder if you have thoughts on, you know, with the two different organs, you've got a different complexity of cell types, and is it, how much does it complicate the work in kidneys with this approach where you've got to have differentiation of so many different cell types and so on?
Yeah. Well, I should have acknowledged Doris Taylor, because she pioneered this. All our work is based on her work. I met with her and Harold Ott, who did a lot of the pioneering work, and it's amazing. So yeah, it's interesting. So she does heart, and it's probably one of the more difficult organs along with kidney. One of the easiest is actually the lung and the liver. The liver, because there's fewer cells, and it's very easy to decell and recell, and same with the lung. We stuck with the kidney because it's actually very difficult, and everybody else gave up, and there's no competition. So it's actually why we stuck with it. And also we have good collaborators. But what we're finding, and this was really key for us, that the studies we did a few years ago showing that we could put on one cell type, and the extracellular matrix would drive differentiation of that one progenitor cell into all the cells we need. That allowed us to move forward. If that didn't happen, I wouldn't be here speaking to you. So, you know, basically, I wouldn't say we lucked out, but our hypothesis was correct, and nature helped us out by allowing, you know, essentially the ECM will drive that differentiation. So we don't need to worry too much about it. And we've shown this in lung with our collaborators. And we've actually, I didn't show it, but we have preliminary data in the pancreas. We can do the same thing. We can take definitive endoderm, put it on a decellularized pancreas, and essentially repopulate an organized pancreas.
Thank you very much. We have a question from Jean Percy. will ECM turn over from PEG to human ECM, then express blood group antigens, the same as the recipient?
Yeah, so that I'll answer that two ways. So the first part is yes. So the extracellular matrix, and we're just in the middle of doing those studies to figure out how long it takes. But yes, the human cells will eventually degrade the pig ECM and lay down human extracellular matrix, so it'll be 100% human. The blood groups, again, come from the blood and have nothing to do with the kidney. So our idea is if we make the stem cells from the person who needs the transplant, then it will be a perfect match and there won't be any issues whatsoever. And coming back, the kidney is optimal for this because we worked out that it would take us about 6 months to make a kidney for somebody. If a patient came in making their stem cells, doing everything should take about 6 months. That patient can be on dialysis. Whereas if you're trying to build a heart and someone has a heart attack, you have about 6 minutes to make them an organ. So it's a little tougher to do that with something like a heart, but it's very doable for a kidney. To make a matched organ to that person.
Okay, thank you. We have another question from Patricia Gongol on the idea of growing the kidney inside a patient while they are still on dialysis, while it develops. I feel solicited patients' perspectives on how they will feel about that.
That's a really good question, but and I'm a PhD, not an MD, and I do transplants on mice, not on humans. Just want to put that out there. When they do current kidney transplants, they don't actually remove the kidney. They basically leave the old kidney in there and put the third kidney inside. So this would really be no different. So it would just be a matter of their new kidney, which would be hopefully made from their cells, growing properly. I mean, it's not a small kidney getting bigger. It's just the cells will be there. They just have to mature. So, but that's a good question for talking to a group of patients about how they might feel about this.
Well, thank you so much, Dr. Rogers. I think we are running out of time. So thank you for all the questions answered. And and thanks for everybody for attending. We look forward to the next week when we would have Dr. Debo Ademola and Dr. Julian Wigley presenting pediatric transplantation in Nigeria, challenges and progress. So thank you so much, Dr. Rogers, for this wonderful talk.
So I just want to, Lori put it, so thank you, Dr. West. So she corrected me, which I'm very happy she did. So with the ABO question, they are also expressed on organs, not just blood cells. That's my mistake. So coming back to that question, again, if we use a patient's own cells like we're hoping to do, the ABO groups will also be matched. So thank you. Thank you very much.
Very nice. Lovely work.
Thank you.
Fascinating.
Take care.
I appreciate it.